Hi, I'm Morgan Michalkiewicz, and for my biology role model project, I chose Elizabeth Blackburn. Elizabeth was born in Tasmania, Australia on November 26, 1948. She grew up in a family of doctors. Both her mother and father were physicians. Even at a young age, Elizabeth always had a fascination with animals. From jellyfish to tadpoles, she would often spend time picking up and observing these different animals. Throughout her childhood, she also had many different types of pets. This curiosity stemmed from animals and nature and led her to have a strong interest in biology, even as a child. As a child, Elizabeth carried a love for playing piano. There were even points in her life when she wished to pursue a career as a musician. Although she had these aspirations, she realized that she was simply competent in piano and not extremely talented. This further pushed her down a path towards a career in the sciences. Elizabeth pursued an undergraduate degree at the University of Melbourne in Australia. She graduated with an honors degree in biochemistry. Additionally, she had started research in a biochemistry lab during her undergraduate education. Her passion for biochemistry is what initially drew me towards her. I have always been interested in biochemistry, and her research inspires me to continue to pursue it. Elizabeth wanted to attend the University of Cambridge in England for a PhD, but in order to be admitted, she needed to spend a year in a lab doing research. Because of this, she continued her education at Melbourne as a master's student, working in a lab that investigated the biochemistry of amino acid metabolism. After this year, Elizabeth was admitted to the University of Cambridge in a laboratory of molecular biology as a PhD student. Here, she worked with to sequence regions of bacteriophage 5X174, which is a small single-stranded DNA bacteriophage. After her PhD work, she went on to pursue a postdoctoral fellowship at Yale, where she researched ways to sequence DNA found in the terminal ends of many chromosomes in Joe Gall's lab. Elizabeth's main focus is in molecular biology and biochemistry. She especially partakes in research surrounding telomeres and the enzyme telomerase. Some major areas she strives to find more information include what roles telomerase plays within the functioning of cells. Additionally, her lab also addresses the question of whether or not telomerase has other functions outside of polymerizing telomeric DNA. The most recent research paper that Elizabeth Blackburn was on is titled Maternal Pro-Inflammatory State During Pregnancy and Newborn Leukocyte Telomere Length, a Prospective Investigation. The study was looking to reveal the effects of the maternal pro-inflammatory state on the length of a newborn's leukocyte telomeres. Throughout the study, 112 women had their blood tested at 3 different stages in their pregnancy: early, mid, and late. Additionally, researchers took one blood sample from their newborn baby soon after birth. The study concluded that a high maternal pro-inflammatory state was linked to a shorter leukocyte telomere length within offspring. Elizabeth Blackburn has made many profound contributions to science. She helped discover the molecular nature of telomeres. Additionally, with the help of Carol Greeter, she discovered the enzyme telomerase. Along with this, Elizabeth received the Nobel Prize in 2009 for physiology or medicine for her discovery of how chromosomes are protected by telomeres and the enzyme telomerase. Her lab is currently investigating the mechanisms that underlie the impact of the limited telomere maintenance on human organismic processes.